Let us start with pathology. First topic is free radical and cell injury. What is a free radical? Free radical is a molecule with only one electron in the outer orbit. It has tendency to steal electron from another atoms, which can lead to cellular injury. What are the sources of free radicals? Then it has primarily physiological sources of free radicals, which is oxidative phosphorylation, also known as cellular respiration. What happens? Normally, two molecules of oxygen require four electrons to form water molecule. When oxygen get less than four electron, the unpaired electron forms the free radical. Examples are superoxide, hydrogen peroxide, and hydroxyl radical. Another way of forming free radical in the cell is pathological conditions. What happens in case of inflammation by phagocytes, the macrophages and neutrophils engulf pathogen and release enzyme NADPH oxidase, and the engulfed pathogen release enzyme phagolysosome, which is a lysosomal enzyme, and then it activates NADPH oxidase, which causes oxidation and release two electrons. These two electrons carried out are taken by superoxide with the help of enzyme superoxide dismutase, which get converted into hydrogen peroxide. Macrophages also secrete nitric oxide synthase, which kill pathogen by breaking their cell membrane and the cellular protein. They also secrete nitric oxide. This nitric oxide get combined with superoxide to form peroxynitrile free radical. Free radicals are also formed with the UV light or X-rays. When the UV light hit the water molecule, this radiation causes water molecule to lose its electron. Another way is hemochromatosis. When the high amount of iron is absorbed, the buildup of Cu or Fe in the body triggers the Fenton reaction. Fenton reaction is Fe2 plus get combined with hydrogen peroxide forming Fe3 plus and OH minus and OH free radical. This reaction is reversible when Fe3 plus combines with hydrogen peroxide. It get converted into Fe2 plus one proton is lost. So this is a continuous reaction causing buildup of OH minus and OH free radical into the cell, leading to the cell death and tissue fibrosis. Ischemia, production of reactive O2 species, ROS. Reperfusion is when more oxygen is taken by the cell. It reacts with the free radicals and causes cell damage, also known as reperfusion injury. Chemicals and medication also forms free radical. Example, when medications are metabolized by livers, lots of free radicals are developed which cause harm to the liver. In defense mechanism, antioxidants like vitamin A, vitamin C, vitamin E donate electron to neutralize free radical. Glutathione neutralizes hydrogen peroxide into water. What happened? The oxidized glutathione is reduced back to the working state by the electron given by NADPH. When NADPH gives electron, it is reduced to NADP plus, and this reaction is taken place with the help of glutathione reductase enzyme, which uses NADPH as an electron donor. The NADP plus is turned back to NADPH with the help of G6PD enzyme, which is glucose 6 phosphate dehydrogenase. It oxidizes into G6P, which is glucose 6 phosphate. There is metal carrier protein. They binds to the metal ions, help transport and store them. The ions are unable to generate free radicals. Then there are scavengers enzymes which convert free radical to the harmless substances. Example, superoxide dismutase converts superoxide into hydrogen peroxide. The catalase enzyme convert hydrogen peroxide into water molecule in peroxisome, and glutathione peroxidase convert hydrogen peroxide into water in the cytoplasm. Free radical defense mechanism also causes cell damage. Lipid peroxidation. Imagine this is a cell membrane. When a hydroxyl radical attacks lipid molecule, it cuts electron from the membrane. So the membrane takes the electron from the adjacent molecule, and this reaction continues and leads to the cell damage. Oxidative modification of protein causes breakdown of DNA, which can lead to the cancer.